Hello, everybody, and welcome back. We are here to wrap up the last bit of our chapter 13. So we've spent all of the other videos taking a look at viruses, which are those filterable infectious agents made up of a protein coat and a genome of either DNA or RNA of various combinations. So this last slide, we're going to take a look at some other weird infectious things that aren't quite well known, like a bacteria or fungus infection would be. So these are viroids and prions. So viroids are actually plant pathogens. As far as my knowledge goes, there's no viroids that have been found to infect humans. They're small, circular pieces of RNA. So you can kind of see it's interesting. Like here's one here in these really small micrographs. And sometimes you see them as flat, like these little right here being pointed to. They just look like a little line, but they are circular. But so imagine if it's RNA is single-stranded for the most part. If you have a single-stranded RNA with all of the base pairs kind of like this, sometimes it collapses in on itself, almost like a double strand. So that's kind of why some of them look circular and some of them look flat, is because it's kind of gone shoop, kind of stuck, hydrogen bonding to each other. So what do viroids do? Well, viroids just kind of cause some problems for the plant. Not a huge, devastating disease, but if you're a farmer, so here is your normal potato crop, and here's your affected potato crop, right? So this is called the spindle tuber potato, no, what is it? Potato spindle tuber virus, or viroid, this word right here. And so it's just this tiny little speck of RNA that gets itself into the tissues, and you can see it kind of makes the potatoes smaller in size, a little bit spindly, and kind of squished and long shaped. So it can definitely impact you as an agriculturist trying to grow a crop. You do not want to be affected by these viroids. So they're just plant pathogens of RNA, their RNA origins. But they don't have any protein coat around them. They're not anything like a virus. They're just infectious RNAs. The next one is called a prion. So prions are weird because they are none of the above, right? They don't have any genome and they don't have a protein coat. They are actually infectious proteins. They don't have any DNA. And when these were first being studied, people were like, that there has to be some reason, a genetic reason why we have these diseases. Because up until the discovery of prions, all infectious diseases had something to do with nucleic acids, right? So the various types of genomes in viruses, bacteria, fungus, protozoa, they all had DNA or RNA or some combination. So yeah, it was hard to kind of fight that paradigm that all infectious diseases had to have some kind of nucleic acid component. So once these were discovered, it was found that these proteins called cellular PRP are normally part of our cells. We're going to focus on the ones in the neurons because that's where they have their most impact. So here in the cellular PRP, we can just kind of see this arrangement. Proteins are three-dimensionally folded. We have the alpha helices, the beta pleated sheets, and all that kind of organization. In the infectious, where they were finding these neurological disorders, they were finding this prion protein, but it was shaped differently. Instead of having these spirals, we had these shapes here instead of the helices. We had the ribbon-looking shape. So they kind of looked at it three-dimensionally, and they're like, well, maybe that has something to do with these neurological disorders. They're like, okay, so we're finding the affected protein. It's this prion protein opposed to the cellular protein. But how does it replicate? They found, and I'm not sure if I think there's a picture in the book, they found that if you have the presence of a prion protein, all it has to do is to physically come into contact with the cellular prion protein, and just that bumping into that direct contact turns the cellular protein into a prion protein. So it's not like it's replicating itself like one prion protein divides, because it can't really divide, it's a protein, but it causes a conformational change in normal protein, and that's what gives it the infectious part. So what does it do to this nervous tissue? Well, it causes what we call spongiformencephalopathy. Yeah. I got all those syllables. So spongiform basically means it turns your brain into a sponge full of these holes. This is not normal. You're not supposed to have big holes in your neural tissue. And we can see in this picture here between the control and the other brain, it's shrunk. There's lots of reduction in white matter and gray matter. So this is the outcome of spongiformencephalopathy. There's a couple different kinds. You may be most familiar with it being called mad cow disease, because that's a big outbreak that happened in Britain in the 80s and 90s. They had to kill a lot of cows. And the reason for that is because maybe some bad farming practices. There is a similar type of disease in sheep called scrapie. And what some farmers were doing is they would take some of this non-utilized sheep parts like brains and spinal cords, which can't be used as meat. They would grind it up in the cow's food to give them more protein. They're like, hey, let's feed our cows protein because then they'll maybe give better milk or have larger muscles or something for their meat components. But unfortunately, when the cows were eating the sheep neural tissue, they were ingesting the prions. The prions got into the cow's body and the cow's brain, and then that was actually transmitted to people. So there's quite a few people that got infected by these prions by eating infected cow's meat, which ate infected sheep brains. So moral of the story, don't feed cows sheep brains and don't eat neural tissue. And you can't kill these by cooking. So cooking the normal disinfectants or removal of pathogens by heat does not denature these proteins. Incineration, but who's going to want to eat incinerated meat, right? So that is your discussion of prion. I think one of your case studies might have something to do with that as well. Comparisons of viruses, viroids, and prions to bacterial cells. So this is just a final comparison table. I'm not asking you to memorize anything, but it's just kind of cool to make that comparison that we've been spending most of the term talking about bacteria. How does that compare with these other three infectious things that were new to this chapter? Viruses, viroids, and prions as far as size, what kind of nucleic acid, if it even has any, cytoplasmic membranes, ribosomes, all of these characteristics we'd normally associate with living things. How do these new categories of infectious things compare? All right, I think that's it. We wrapped up chapter 13. Way to go, everybody. So we have one more chapter to cover. That's going to be chapter 14, epidemiology and infectious disease. So I will see you next time. Bye.